Welcome to the Pharmacy Inspection Podcast, where we discuss topics related to sterile and non-sterile compounding pharmacy in an effort to promote compliance and increase quality. The Pharmacy Inspection Podcast is a member of the Pharmacy Podcast Network, delivering quality and meaningful conversations and discussions about our pharmacy industry and the critical role pharmacists play in our healthcare systems. Learn more at pharmacypodcast.com. Please welcome your hosts, Brian Prince and Seth DePasquale.
Hello to all of our compounding friends and colleagues, and welcome to the Pharmacy Inspection Podcast, where we discuss all things compounding in an effort to further the cause for quality and safety in the compounding industry. You are listening to the first and original podcast series in our industry, and we will continue to push our educational and knowledge-sharing agenda with you, our audience. So with that in mind, I ask this week's guest, Dr. Tom Kupiec, to join us because he is a very knowledgeable gentleman with a lot to say and a lot to share with compounding pharmacists. So last week, I was in Oklahoma City, and I was at a conference, ARL Quality Conference, and I have to say that it was one of the best conferences that I've been at in a long time because, to me, there was a wonderful crosshatch of folks from our industry that had a lot of good and great different perspectives. And every session that was there was just action-packed with good information, technical information, information that you could just take away and feel. And so I wanted to circle back and actually invite the host from ARL. His name is Dr. Tom Kupiec. And I know he has a lot of wonderful things to say. And so just as kind of a precursor, we had Jenny Sun from USP, Kim Kiefer from Empower Pharmacy. She's actually been a two-time guest. She was on with us again last week. Ken Latta, Dr. Lloyd Allen, a gentleman by the name of Tony Park I'd never seen speak before, who was a lawyer and both the pharmacist, gave a great perspective. But this week, we want to welcome Dr. Kupiec to come in and kind of backfill some of the things that's going on. But before I get started into what we listened to at the conference, Dr. Tom, if you would please give us some of your background and how you got to this point in your professional career.
Well, my background, I started out working for the police department as a forensic chemist. And then I had training at the FBI, DEA as part of the training at the forensic laboratory, but actually testing drugs or illicit drugs. From there, I went to work for the Federal Aviation Administration, Department of Transportation in the toxicology laboratory. So I performed testing on samples from the airplane crash accidents. And so essentially, after working at the police department, I ended up going to work for the forensic toxicology area in the Department of Transportation, Federal Aviation Administration. And while there, I went back to school and obtained my doctorate in pharmaceutical sciences from the University of Oklahoma Health Sciences Center, College of Pharmacy. And so the PhD was in pharmaceutical sciences. And after leaving Oklahoma, I went to Salt Lake for a period of time and then came back and acquired ARL in 1998. And since then, this is our 20th anniversary, and it's been 20 years of steady growth. We've averaged double-digit growth every year for 20 years.
I appreciate you sharing that with us. So I want to kind of backtrack to where we were last week at your quality conference. And I appreciate the fact that you and your company were willing to host that, because like I said, there's just a great crosshatch of folks that came. So if you could summarize, give us some of the high points, some of the takeaways, some of the things that we heard that you want to share with our audience.
Sure, I can do that. It might be good just to say how the idea started, because frequently we're asked this question, how did it begin? It really was a thought of discussion in the hallway at Compounders in Capitol Hill, the IACP Compounders in Capitol Hill meeting in the summer in Washington, D.C. And that meeting is often focused on regulation and lobbying and working at the national level in behalf of compounders. And our business really has to do with quality control and quality testing and ensuring the quality of compounded products. And we were talking to Jeannie and Dr. Allen and Joe Caballera in the hall, and it said it would be nice to have a a meeting that focuses on the science or the technology of compounding. And they said, sure. So over a couple, 3 years, we talked about different topics that might be beneficial. And that's really how it came about. The Quality Compounding Summit is a brainchild of multiple people. I can't get the credit for that. This is great people at USP, such as Jeannie and Rick Schnatz, and then Joe Caballera and Ken Lotta. And there's many, many people that are to be thankful, IACP, to be thanked for the reason for this. ACA, it's hosted, it's co-hosted by ARL and ACA. They accredit it so the individual attendees can get continuing education credit for their time at the summit, the Quality Compounding Summit. But that's how it really began. You know, you were asking what are some of the takeaways, and maybe I can go through just each of the speakers and highlight a few of those. We'll start out with Jeannie Sun. You know, Jeannie, she is actually, her talk was on updating the development and revisions of the USP compounding chapters. And there's tremendous interest in the USP 795, 797. And Jeannie did a great job of giving an overview of the 795 non-sterile preparation chapter, as well as 797, the sterile preparation. And then there are many questions today and even forthcoming on USP 800, the hazardous drugs chapter. And I think one of the things that big takeaway, and the reason why USP is so good is that it's a committee driven, and they have asked, and she has asked for compounders across the nation to invited them to call in to submit electronically through the USP website their questions or their comments on the 797. And I bet that came across through numerous times throughout our talk, wanting input from the stakeholders in compounding. So this is a time to put your comments in, to address the concerns or questions that you might have on the 797 and 795 that's out there. Ken Lotta, he spoke on controlled environments for sterile and non-sterile compounding. And I think why Ken is such an incredible speaker is it's interesting because each of these speakers have their talent and their knowledge and skill, and they've been around, and they're considered leaders in their field. And I think that Ken, not only is he a pharmacist, but he is very good with design and regulatory compliance. And so Ken spoke a lot about 795, 797, and 800, and a lot of his time was discussing facilities and design. And he talked about the proposed 797 and some of the issues with it, the temperature and humidity requirements in USP. He also discussed some of the concerns or common issues with certification providers. And then one of the, I thought, seminal comments, he said, people are the source of contamination, which we know. I think he mentioned in one of his slides that there was 100,000 particles per cubic feet per minute that the person just sitting motionless, 2.5 million particles per cubic feet per minute, a person sitting down or standing up, and then 10 million particles per cubic feet per minute if a person is walking. So you can see that concept or the idea that he was trying to get across, that really the people are the source of the problem or the contamination. And that was, I thought, a very interesting comment that he brought up during that. And then he presented another aspect that Ken did was showed numerous designs. And then he mentioned the design of the unilateral direction that the FDA tends to favor. And I think there's many designs that are operable, that are appropriate. And I think it's important to get experts when designing a clean room, such as Ken and many other good vendors out there that provide those type of opportunities. I spoke actually on the scientific and technical challenges, and Kim and I sort of teamed up in that I gave the scientific, basically challenges that we've seen in the lab over the years. And then when you're looking at the out-of-spec investigations, we're trying to do a root cause analysis or look at that root cause of that issue, you're looking at that scientific or technical challenge that could have contributed or caused that out of spec. And I think when you're looking at the foundational issues, you can solve them more mechanistically or for long term, not just put a Band-Aid on it and treat the effect, but really treat the cause. So I focus on the science and the technology of many of the issues that we see in compounding today. And some of the things that we talked about just in general were like salts. We know we gave the example of fentanyl citrate, how it may be labeled as fentanyl citrate, but the concentration is actually 50 micrograms per mil of fentanyl. So, and then, and showed how to do the calculations and gave some examples like that. Another topic which presents a lot of problems we're seeing in compounding today is the water issue. And from an example from hygroscopic term to hydrates, where a hygroscopic might be that drug product, physically could be grabbing or water seeking from hygroscopic, so you may have moisture in there too. Hydrates, where there's actually a molecule of water that's integrated into that molecular structure for something, such as a hemihydrate or a pentahydrate. And we gave several examples of that for when do you include the calculation, how do you deal with a hydrate in the formulation. And as Kim brought out, and we had a lot of comments that they enjoyed her talk because it gave practical perspective and practical application for the compounding pharmacist. And really, it gets back to the basic pharmaceutics that you learn in pharmacy school, or the calculations. I mean, the calculations on dealing with a hydrate or a hemihydrate or a salt, when to consider it, when not to consider it, those were examples that were heavily focused on at this particular meeting this year. I also spoke about blending and overblending and segregation of the particle sizes. Particle sizes is a big issue. We spoke and gave some examples of filter binding, how you would perhaps create a formulation and then run it through a filter, but some of the active ingredient may bind to that filter. And often people would wonder, well, why is it so low? And so that process we would look at, and we gave some examples of the filter binding issue. Other issues were the light and packaging and content uniformity. We discussed the content uniformity and gave examples of several creams and ointments and how over time it could segregate or separate, and that's what we don't want. The desired outcome is a pharmaceutically elegant product or a preparation that is given to that patient that is safe and efficacious. We ended up my talk on, I wanted to really dive deep into one particular area, and so since there's a lot of questions on container closure integrity, we talked about the issue with Avastin, and we gave some examples of Avastin and how the proteins are folded, and the protein and packaging has created some issues of aggregation and with the Avastin in the syringes, and then ended up with some citations of several of the peer-reviewed scientific journals talking about the contamination of the anti-VEGF drugs for intravitreal injection or silicone oil microdroplets and protein aggregates. So when you looked at the science, what's causing that oil to react with that Avastin in that syringe, and we discussed those issues in this particular meeting. And then we ended up with some basic examples that Kim presented specifically of some of the scientific products that I had brought up. And one of the, I thought, most important, Kim was saying, was read the USP monograph. And what does that monograph actually tell you? For instance, the drug substance, it tells us the assay, the purity, the activity, what it should be, the chemical formula, the water, the loss on drying, the residue on ignition. I mean, there were many issues that are brought up in the development and the information that's listed in the monograph, in the drug monograph. Whether you go into the USP, the Remington, or other references, those are invaluable. And so Kim talked about those. Again, she further discuss the water determination, such as in Chapter 921, or the titration, as I mentioned, and versus the loss on drying, which is actually, we were differentiating water determination as looking at the water in that product, whereas the loss on drying does not only include the water, but it may be other organic volatiles. And so when you perform the 731 USP, you're looking at not just water, but the loss of volatile matter that might be related to that particular substance. She talked about labeling. We talked about calculating and formulations. And this actually started from a situation with atropine sulfate monohydrate. That was an example that when you looked at it, in different instances, at one point you should consider the hydrate or account for it. Next time you would not. Another time you would look at the salt. And then so I think this is an example from the information that was given at that Quality Compounding Summit. And I think people think, well, why do you do this? I mean, it's so important to ensure for compounders quality, and that's the whole reason for ARL. I think that testing or those quality purposes provide education, technical support, and network with other industry providers.
So, Tom, I had the pleasure of going to dinner that evening with your team, and actually everyone from the conference was invited, which was just wonderful. But I wanted to let you know that everyone on your team, and I got to sit beside actually three of them across the table and beside me, and they're just wonderful, pleasant attitudes. You could tell they just genuinely enjoyed what they did. So I commend you on your team. And I have a very simple business theory. It's culture, customer, cash flow. And to me, the business is always built on the culture. To me, that is the foundational principle for which you're building your business. So I want to commend you that I can see that you're building inside of your company just a wonderful culture. So I appreciate that. But, and we don't usually turn this into a commercial for anybody, but because your company stepped out there and decided that you were going to actually host a conference and bring as much wonderful technical information, and really everybody was playing off of everybody else. It's like Dr. Allen was synergistic to Jenny. Jenny was synergistic to you and Kim. And it's like, I mean, everybody was just playing off each other. And it just, to me, it built this better mousetrap. So I tell you, with saying that, tell me how ARL, how do you guys partner with pharmacists to help raise the level of quality? Because you keep using that word quality. And I genuinely appreciate that, because right now we're at a place where, we're concerned with even the new, proposed revision of 795 is, longer BUD studies. And we've always been concerned about stability studies and endotoxins. And those are always the things that we talk about. So tell me how your company partners to help build quality for a pharmacy.
Well, I think it's paramount that the whole purpose is to deliver a compounded preparation to that patient, and to make sure it's safe and it's effective and it's the appropriate dose, it's the appropriate aspect of it. And that all centers around quality. And it's difficult sometimes to define quality without using the word quality, but that includes knowledge and skill of preparing that substance. It ensures following the compliance aspect, the testing, providing education, and understanding the scientific and technical aspects of compounding, which gets to the root basis of compounding in the pharmaceutics aspect. And I think that ARL wants to partner with pharmacists and others and big pharma and individuals, whether they're a 503A, they're a 503B, or they're at the GMP level, to produce a safe and effective drug preparation or product.
Dr. Tom, we certainly appreciate you joining us on the Pharmacy Inspection Podcast. Your information has been great. And I would encourage you folks that if you're looking for educational opportunities going into next year, into 2019, and you need CEs, because this was a C credited through the ACA, then I highly, highly recommend that you join this conference next year. It'll probably be around the same time. Was that around the 1st of October, Dr. Tom? Is that about the same time we're going to host it again next year?
Yes, it's going to be either in September or October. We work hard at not to interfere with the other shows to maximize that time and try to be aware or cognizant of the time for the pharmacist or the pharmacy technician.
Yeah, and I'm sure they appreciate them. After there was a lot of people that, they were up and leaving and heading over to the NCPA, and there's always, these late season conferences. So, last opportunity, any following or any last pieces of advice that you want to toss out there? We always like to leave our audience with one more just good nugget of information.
I just think that quality elevates excellence, and I think that rising tide floats all boats, and it's really important to have a quality compounded product or preparation given to that patient. And I think foundational, and there's so many people that contribute, and we consider it an honor to be able to serve this community.
Wonderful comments. Again, Dr. Tom, we certainly appreciate your time on the Pharmacy Inspection Podcast. We hope to hear from you soon, and don't, always feel like you can be a regular guest for us.
Thank you.
That's it for us. Another great episode of the Pharmacy Inspection Podcast. A special thanks to Tom Kupiec from ARL. I really appreciate this conversation. It's been a great talk, and allowing us to pick your brain. To be totally honest with you, I have used ARL on many occasions as an extension of my own pharmacy, and continue to do so still today. They're great. One of those great reputable labs. One of those great reputable labs that you can count on. I definitely encourage anyone, if you're looking for a lab, I don't think you can go wrong with ARL. They're really a good group of people. Special thanks to who wasn't on the podcast, but Todd Lynch personally has helped me out a lot over the last several years, and thank you. Coming up this week, though, I did want to mention Brian and I are going to be putting on a webinar about USP 800 hazardous drug flow, containment strategies, and how to work with hazardous drugs. It's going to be a great webinar, really informative. I've seen this webinar at different conferences before, and I know that it's not going to disappoint, so I encourage you to sign up. Thanks to all those that have already signed up, and we really appreciate it. So yeah, thanks again to everyone, pharmacy friends, colleagues. Keep raising the bar.